Transgenic plant. Transgenic plants are plants that have been genetically engineered by using DNA recombinant technology to create GMO plants with new characteristics. Genetic modification is a technology that involves inserting DNA into the genome of an organism. To produce a genetically modified plant, new DNA is transferred into plant cells. Usually, the cells are then grown in a tissue culture where they develop into plants. The seeds produced by these plants will inherit the new DNA.
Canola, sugar beets, alfalfa, yellow squash, zucchini, Hawaiian papaya, cotton, corn, soy are some of the examples of genetically engineered foods.
What are the requirements for transgenic plants? What we need are basically plant cells, vector, DNA of interest, enzyme to cut DNA, and bacteria, for example, Agrobacterium tumefaciens.
Next is on how to produce transgenic plants. One of the ways is via Agrobacterium tumefaciens. It is a soil-borne gram-negative bacterium with root-shaped and form water in rhizous form. These bacteria are commonly known to cause crown gall disease in their cotyledon plants. Besides, these bacteria also are attracted to one site via chemotaxis. Chemotaxis is the movement of organisms in response to chemical stimulus, and in these cases, the bacteria are attracted to sugar and phenolic molecules released from damage of plant cells. In addition, scientists found that these bacteria are able to transfer bacterial genes to plant genome. And lastly, these tumefaciens also contain PR plasmid, also known as tumor-inducing plasmid, that can transfer its cDNA region into genome of host plants. Next is steps in production of transgenic plants. The process of plant modification usually use bacteria named Agrobacterium tumefaciens. This bacteria is characterized by the presence of PR plasmid in its cell. This plasmid contains genes that are responsible for the development of crown gall disease. It appears as a tumor-like swelling on plant root. In laboratory conditions, it is possible to replace this gene with another gene that contains desired properties. In the first phase of plant genetic modification, young plant leaves are cut into tiny pieces before being exposed to plant hormone. Then, into the leaf that had been exposed with plant hormone, Agrobacterium tumefaciens will be added. At this stage, the desired gene is transferred from the plasmid into the plant cell genome. With the presence of plant hormone, a single plant cell that had been transformed by the plasmid developed into an entire plant possesses desired trait.
And I'm going to proceed with the examples of transgenic plants. The first example is bud-killing cone. Bacillus thuringiensis was first discovered by Shitegane Ishiwatari while investigating the death of a mass of silkworm caused by Soto disease in 1901. Ernst Berliner then rediscovered the bacteria and named it. Bt-delta endotoxin produced by the bacteria is capable in killing Lepidoptera larvae. Bt has been used widely as an alternative of insecticides. Moreover, it does not harm other insects, for it is very selective. The second example is non-browning apple, or also known as Arctic apple. Canadian scientists came out with a product, apples that have an ability to resist browning after being cut. This is done by silencing the gene that is responsible in making the enzymes that causes the browning in apples. The enzyme, known as polyphenol oxidase (PPO), reacts with oxygen and polyphenols when apple is cut and leads to apple browning. By discontinuing the production of PPO, it slows down the rate of browning and aging of the apples. The differences in compositional analysis between Arctic and conventional apples are small and would not adversely affect the nutritional quality of the apples or raise any safety concerns.
Next, we move to the third example of transgenic plants. The third example is Flavor Saver tomato. Flavor Saver tomato is the first genetically engineered crop granted license for human consumption. It was produced by Californian company Transgenic in 1992. Transgenic introduced a gene implant which synthesizes a complementary mRNA to polygalacturonase PG gene and inhibiting the synthesis of PG enzyme. On May 21st in 1994, the genetically engineered Flavor Saver tomato was introduced. This was achieved by adding another gene that blocked the gene for the production of an enzyme which causes rotting, polygalacturonase. Due to various commercial problems, the tomatoes are no longer available. I will explain about the gene, which is Flavor Saver gene. Flavor Saver gene is a strand of RNA that is reverse of the RNA that occurs in the plant. Original RNA produces enzyme polygalacturonase (PG). Flavor Saver RNA will bind to polygalacturonase, so the enzyme cannot break down pectin. It will cause tomato to become soft. The two strands cancel each other out. Making of Flavor Saver. Enzyme polygalacturonase breaks down structural polysaccharide pectin in wall of a plant. This is part of the natural decay process in a plant. Flavor Saver tomatoes have been constructed that express an antisense mRNA complementary to mRNA for an enzyme involved in ethylene production. These tomatoes make only 10% of normal amount of enzyme, thus delaying ethylene production. Normally, tomatoes are picked while green and transported many miles before being sprayed with ethylene to ripen them. This prevents damage and perishing on the journey. Basically, the gene in the tomato stops the tomato from softening during ripening, so that it is easier to ship, but keep its natural flavors too. The tomato also has a much longer shelf life and keeps from spoiling quickly. As you can see, the image shows three sets of tomatoes. The ordinary control tomatoes soften and shrivel up, while the texture of gene-silent tomatoes remains intact for up to 45 days.
Hi! Now, let's talk about the advantages and disadvantages of transgenic plants. Let's go! The advantages are pest resistance, overcoming malnutrition, virus resistance plants, and increased productivity. Crop losses due to pests means huge financial loss. The pesticides were introduced, however, remain not safe for humans. Hence, the Bt toxin gene is transferred to the interest plants. The third world countries are facing extreme malnourishment. Some crops, like rice, has inserted with vitamin A to avoid the deficiencies. Plants can be resistant to diseases. As an example, viral crop proteins were introduced to tobacco and tomato for resistance to tobacco mosaic viruses. Transgenic crops can produce more yield than traditional crops. This is because the reduction of losses by pests, viruses, and weeds that compete for soil nutrients indirectly increase the final yield. The disadvantages are allergies, ethical issues, and risk of harming other organisms. The imported genes produce a new protein which has never before been present. The novel protein may trigger the allergic reactions. Some ethical issues are inserting animal genes to plants, which is against some religious views, and not putting labels on food by the manufacturers. The studies say that the pollen from baby corn cause the high death rates of monarch butterfly. There are a lot of benefits of transgenic plants, but in addition, it may produce a number of threats too. Hence, the world must ensure that the standards are followed and being continuously monitored. Thank you for watching.